Good day, everyone. I am Proceso Perturbos III, and together with the team, we'll be talking about evolution and development. It cannot be denied that evolution is the central theme in biology. Evolution and development had always walked side by side in the course of time. There are three things that would help us untangle the relationship between evolution and development. They are macroevolution, the tracing of the primordial animal, and the question of what really happened in evolution. Before we start, let's have a short activity. Try to guess the word from the clues and jumbled letters that we have provided. The answers will be given as we progress in the discussion. Feel free to pause and play and try to guess the items. For the first item, we have here three pictures. The first are a set of birds, which is the work of the third picture, and the second one looks like a phylogenetic tree. For the next item, what looks like a shell on the first picture and the fish on the second, as well as a very familiar button on the third. For the next item, we have here planes and axes, as well as an outline of a human and some parts of the insect. The next is a house, a container, a DNA, and what looks like a spinal cord. For the next item, it looks like a front and back of the human and an insect. For the next part, we have what looks like a crustacean and a bee with its head, thorax, and abdomen. Very simple or familiar word. And for the last part, we have here what looks like a human development with different body parts having significant changes. Same goes with the crab, its pincer and antenna having significant changes. All right. So to start with our discussion about evolution and development, we'll start with macroevolution. Macroevolution is that would help us untangle its relationship with developmental biology. And they are namely molecular taxonomy, phylogeny of animals, and fossil records. In discussing evolution, it is inevitable to discuss taxonomy. Like from the highest levels, like the domain, kingdom, up to the lowest, like the genus and species, we refer with each level or to each level as a taxon, if you can remember. For the animal species, they are grouped into genera, family, order, classes, and even the highest grade taxa are in the form or in the phyla level or the phylum level. Furthermore, if you can recall, each taxon is a clade consisting of the complete set of descendants from a common ancestor. This is important to have a backtrack or a review on this because as we look into basal and derived characteristics, we tend to focus on certain taxa which could actually be a clade in itself. Going further, as we are discussing with macroevolution, it's high time that we try to contrast it with microevolution. As we know, evolution is the process of how living things change over time. It can happen on a small scale or a big scale. So when we talk about small changes within a population, we call it microevolution. And this can happen very quickly. Like there's a frequency of a gene for brown coloration in beetles increasing in just one generation. So that's microevolution because it's in small scale and the level of the species. Microevolution rely on mechanisms like mutation, migration, genetic drift, and even natural selection. On the other hand, macroevolution is about changes that happen over a longer period of time, and they can result in the creation of a new species. For example, the evolution and radiation of the dinosaur lineage is an example of a macroevolution, as we can see here in the picture. So we can't see macroevolution happening directly, but we can study it by looking at the diversity of life over time. And As we study, it includes evidences such as fossils and even living things. So just to wrap up, microevolution happens with a single population and can't be beyond a single species. And it's observed over short periods of time, while macroevolution happens on a bigger scale and can result in the creation of a new species. Both rely on the same mechanisms of evolutionary change. And when we study macroevolution, we look at multiple lines of evidences. In relation to evolution and developmental biology, we look into homology and analogy to describe similarities in the different organisms, or rather in the development of different organisms. So homology here refers to similarities in the developmental patterns that are shared by organisms or shared between organisms because they have the same or they have this shared ancestry. Like say for example, in our example here we have the human hand, the cat's feet, the whale's flipper, and the bat's wing. Even though the limbs themselves are different in terms of their function, they share a similar pattern of development because the developmental processes that form the limbs or the limb buds are controlled by similar genes and are inherited from a common ancestor. Analogy, on the other hand, refers to similarities in the developmental process or patterns that are not shared due to their um, ancestry, but rather they are due to convergent evolution. Like say, for example, we have here the shark's fin, the penguin's wing, and the dolphin's flipper. They have similar function, however, they are not from the same structure because they develop through different processes and are controlled by different genes. They evolved separately due to the similar demands of their environment. So in biology, we often use the term basal and derived to describe characteristics of the organism. And these terms are actually closely related to homology and analogy. As we all know, basal characteristics are those that are shared by a group of organisms that are considered to be primitive or ancestral. So these characteristics are often homologous, meaning they are similar due to their shared ancestry. So for example, we try to give like the fish and other aquatic organisms being or having gills. And that is its basal characteristic because it is shared among many different groups of aquatic animals and it is believed to have evolved early in their history. On the other hand, we have what we call as derived characteristics, which are quite unique to a particular group of organisms, and commonly we regard them as more recently evolved. Looking at the example that we have here, say for example, if we refer to the vertebrae, among all the organisms here, we can say that the vertebrae is a basal characteristics. However, if we say for example, hair, the hair part is actually exclusive to the primates and the rabbits group. So that taxon has what we call as a derived characteristic. In the same way, if we look at the eggs with shells, which is exclusive for crocodiles and birds in this example, we may refer to that as a derived characteristic. So to sum up everything, think of macroevolution as a skyscraper and developmental biology as the blueprint that guides its construction. The skyscraper is the end result of many small-scale construction activities, just as macroevolution is the result of many small-scale evolutionary changes. The blueprint provides the instruction for how to build the skyscraper. So just as developmental biology provides the instruction for how to build an organism. And in the long run, when there are many organisms that are being built, we tend to see it in a bigger picture that relates now to macroevolution. Because as mentioned, just as the blueprint for skyscraper is considered to be a set of detailed instruction that guide the construction, Developmental biology provides the instruction that guide the development of an organism from a single cell to a complex multicellular being. And just as the skyscraper can be modified or expanded over time, the instructions in the blueprint can also be modified or adapted to suit the needs of the construction project. At this point of time, I will now give the floor to the next reporter, Ms. Yana Manius.
Hey there. Let's chat about the evidences of macroevolution, namely molecular taxonomy, phylogeny of animals, and fossil records. Did you know that scientists now rely on the primary sequences of genes rather than the physical characteristics to classify different species? Genetic sequences changes gradually over time, but most changes are neutral and don't necessarily impact survival or the reproduction of the organism. However, sometimes mutations can spread and can become the norm within a species. Hence, both morphological and molecular attributes are helpful in classifying animals and other organisms. Fossils, too, can help us in setting timelines regarding how the animals today came about. Hi, I'm Ayana Munoz, and this is my section of this report. Let's go to molecular taxonomy. Molecular taxonomy has greatly expanded our understanding of homology and analogy. Even though neutral mutations causes changes in many amino acids, certain positions retain the same amino acids, which indicates homology between genes in different organisms. But things can get tricky with gene duplication as one copy can have a new function while the other retains the original function. When genes result from duplication within the same organisms, they're called paralogs. When genes in different organisms share a common function, they're called orthologs due to lineage. It's crucial to distinguish true orthologs for accurate species comparisons. And finally, we have analogous genes, which can converge to the same function from different sequences. Lacking sequences identify except in the critical region responsible for the biochemical activity. A great example of this is the crystallins in the vertebrate lens, where diverse proteins are employed to create a transparent lens. Even though these different types of proteins don't share any common primary sequence, they all work together to fulfill the same purpose. Moving on to the phylogeny of animals. To construct phylogenetic trees, scientists compare genetic sequences using algorithms that minimize substitutions or differences. Certain genes, like ribosomal RNA genes and some protein coding genes, are ideal for these studies because they exhibit homologous traits and variation without too many substitutions. The consensus of phylogenetic trees represents key animal groups, and due to the lack of shared morphological characteristics among adult animals from different phyla, embryonic and larval information has been traditionally used to construct the tree. One important characteristic used is the number of germ layers. With most animals being triploblastic, meaning they possess three tissue layers, namely the endoderm, the mesoderm, and the ectoderm. However, diploblastic animals such as your porifera, also known as your sponges, your cnidarians under the groups of hydra, jellyfishes, and sea anemones, and ctenophores, also known as your comb jellies, have only two layers and show radial symmetry. The presence or absence of a coelom, also known as your body cavity, is another significant feature. Some invertebrates lack a coelom, and these are what you call your acoelomates, while others have a partially surrounded body cavity, which are called your pseudocoelomates. Coelomate phyla were traditionally divided into two groups, namely Protostomia, and these are organisms that have their mouths formed first, and second, your Deuterostomia, with organisms having their anuses formed first. These are also supported based on characteristics like cleavage pattern and coelom formation. However, molecular taxonomy has led to a revised classification with Deuterostomia remaining as a taxon and Protostomia now divided into two other groups, namely Lophotrochozoa, including your annelids and mollusks, and Ecdysozoa, including your arthropods, nematodes, and other molting organisms. Moving on to the fossil record. The fossil record does provide limited evidence of ancestral relationships in phylogeny, as it is challenging to determine if a fossil represents a direct ancestor of another fossil found at a higher stratigraphic level. However, the fossil record does offer valuable information on the timing and the appearances of different groups, providing calibration points for phylogenetic trees. First, we have the Cambrian explosion, which occurred over a relatively short period of 5 to 20 million years. Short. The Cambrian explosion marked the appearance of most invertebrate phyla. Constructing phylogenetic trees based on modern organisms' characteristics becomes challenging due to the rapid diversification because of this explosion, where the majority of evolutionary changes took place. Second, the fossil record also aids in establishing a coherent chronological sequence of geological sites and provides the basis for the geological timescale. And finally, we have the Ediacaran period preceding to the Cambrian period, which is characterized by fossils resembling Nidarians. Although uncertainties remain due to the challenges of preserving jellyfish fossils, and some metazoan animals have been found during the Precambrian rocks, such as small worm-like creatures in the Deschantual Formation, indicating the presence of coelomatic animals with a thorough gut and anterior sense organs. The phylogeny of animals and even the fossil record can be compared to a giant family tree that represents evolutionary history of all animals on Earth. At the top of the tree, you have the oldest and the most ancient ancestor, which represents the common origin of all animal life. And as you move down to the tree, the branches start to form, representing the different groups or categories of animals, similar to a human family tree. Each generation represents a group of animals that share a most recent common ancestor. For example, one branch might represent mammals, with sub branches representing specific groups like primates, carnivores, and even rodents. These sub branches would further divide into smaller branches, representing more specific groups like humans, cats, and mice. As you continue down the tree, you'll find more branches representing other groups like birds, reptiles, amphibians, and even fish. Each branch represents a distinct lineage of animals that evolved over time. Overall, the phylogeny of animals show a different groups that are related through common ancestors and helps us understand the evolutionary history of life on Earth and how different species are connected to one another. That is my segment of my report. Moving on to Kenneth's.
Previously, we have just learned about the phylogenetic tree and the fossil record, in which homologies can be observed on members belonging to the same taxon. Now, in this part, we will talk about the body plan, the phylotypic stage, and the zootype. So, first things first, the most important novel result of evolutionary developmental biology is the ability to uncover previously invisible homologies. So these invisible homologies are actually the likeness in structure, which are the result of a descent from a common ancestor. So these have been discovered through the study of expression and function of the genes that make up a body plan. So here we can see pictures of different animal body plans, such as the acoelomate, which don't have a cavity, and the pseudocoelomate, which have a false cavity. You also have the asymmetrical, radial, and bilateral. But before we go deeper into that, let us first talk about what really is a body plan. So now we may pose the question, what is a body plan? So a body plan actually refers to the idea that it is possible to abstract the essential features of anatomical organization from a wide range of animals. We've actually been able to observe body plans in our developmental biology lab just recently when we were able to view the body plan or the structures, anatomical structures of the pig embryo, which is also observable in other mammalian embryos. This is also done in zoology textbooks whenever they show a diagram of a generalized vertebrate or mollusk. So this is what we have also been doing in our invertebrate zoology laboratory when we are able to view the generalized body plans of different phyla. Evidence that the body plan is real rather than an arbitrary or random abstraction has come from the developmental biology because of the fact that key transcription factors and signaling molecules are found to have very similar expression domains within a taxon. So, for example, the expression of the brachyury gene in vertebrates occurs in rather differently shaped regions in zebrafish, xenopus, chick, and mouse, but they all correspond to what was previously thought to be the mesoderm. And indeed, they reinforce the concept of mesoderm as a real cell state definable by the activity of a combination of transcription factors and not just an arbitrary label for an embryo region. How evolutionary developmental biology played a role in discovering more about the developmental processes that are similar and can be observed in animals of the same group or taxon is really significant. So, to better understand the concept of body plan and how it is being observed or manifested, we'll now talk about the phylotypic stage. So, the phylotypic stage is the stage of development at which all members of a taxon show the maximum morphological similarity. So basically, at this point in their development, those that belong in the same taxon look very similar with each other on their anatomical structures. For example, insects all look similar at the extended germ band stage. So at this stage, the segmental arrangement of the body of insects is very obvious. For example, they all have three nasal, three thoracic, and a variable number of abdominal segments. So the extended germ band is reached before dorsal closure of the embryo and consists of a plate composed of ectodermal and mesodermal components. So for example, there is a ventral nerve cord, but most details of the future epidermal structures are yet to appear. And the endoderm in terms of the anterior and posterior midgut invaginations has yet to form. On the other hand, among vertebrates, the phylotypic stage is the tailbud stage. It is when all vertebrates have a dorsal nerve cord with anterior specialization, segmented somites, a ventral heart, and a set of pharyngeal arches. So the tailbud stage of the vertebrates can be seen on the picture beside. So we can see the brain, the somite, and the tailbud, and other anatomical structures which can be observed or similarly found in other or most vertebrates. Phylotypic stages can also be observed in groups other than the insects and vertebrates. So, for example, it can be observed in the villager larva of mollusks. So, in this stage, the villager larva of mollusks has a head, a posterior, a through gut, a dorsal shell gland, and a ventral muscle foot. Aside from that, a phylotypic annelid might be considered to be at a stage at which a certain number of segments have arisen and the organism has a head, a ventral nerve cord, a through gut, and segments involving all the germ layers. Also, the Nopleus larva, which has three pairs of head appendages, has been considered as a phylotypic stage for crustaceans. So the said phylotypic stage, again, is the point in time in their development in which these said structures can be found in the members of the said taxon or group. After the discovery of the phylotypic stage, there have also been identified as zootypes in animals. So first things first, it is important to note that the discoveries of developmental biology make it possible to compare the expression patterns of key developmental genes between phyla. So, what is a zootype? So, it is actually the totality of common expression domains that is found on body plans or that is expressed during body plan formation. Also, the zootype, now more appropriately called the triplotype, is a common constellation of expression domains. So, this set expression domains can be shown in the picture in the next slide. So, now in this figure, we can see first at letter A the transcription factors active at the phylotypic stage. The HOX-A and HOX-P indicates a set of HOX genes with nested expression domains having different anterior limits. On letter B, we can see homologous dorsoventral signaling systems in insects and vertebrates. So basically, these expression domains are responsible for the expression or the formation of the said body plans that can be observed on the animals that belong to the same taxon or group. Now, to better understand the concept of the phylotypic stage, the zootype, and most especially the concept of the body plan, we will now proceed with our analogy time. Imagine you're building a house. Before you can start constructing the walls, roof, and other parts, you need to have a blueprint or a plan that outlines how the different rooms, doors, and windows will be arranged. This plan serves as a guide for the construction process and ensures that the final product is functional and aesthetically pleasing. In the same way, a body plan is like a blueprint. So this is the analogy for the or representative of the body plan. It is a blueprint for an organism. It describes the overall organization of the body, including the number and arrangement of limbs, organs, and other structures. This plan serves as a framework for development and ensures that the final organism is able to function properly and carry out its necessary biological functions. Just as a blueprint can be used to construct different types of houses, there are different body plans that can be found across different organisms. So we have actually mentioned this in our previous talk, just like how there are body plans for insects, for vertebrates, and other groups such as the villager and mollusks. So, for example, again, in humans, they have a bilateral body plan with two arms and two legs. So, this is actually observed in all humans that are arranged symmetrically on either side of the body. So, in contrast, some animals have a radial body plan with multiple arms or tentacles arranged in a circular pattern around a central axis. Overall, the body plan is an essential aspect of an organism's development and survival, just as a blueprint is essential for building a functional and sturdy house. Before we come to a conclusion on my part of the report, I will first answer what we have just presented on the first part of this video. So basically, from what we have learned, the answer to the question or the four picks one word is actually a body plan. Before we end this part of the report, I will leave you this question in order for you to be able to recall and look back on what we have just learned. So, this is the question. It is a theorem stating that it is possible to predict the key components of anatomical structure from a variety of species. That would be all for my part, and thank you for listening.
Now, before we proceed to the next topic, I'd like to ask if you got the answer to this question. So, did you get it? The answer to this one is homeobox. So, our new topic is about Hox genes, which are the genetic choreographers that bring our body plans to life. So, what exactly are Hox genes? It is a type of a homeobox gene that plays a crucial role in the anterior-posterior patterns exclusively in animals, specifically bilaterians. It is said to have occurred around the time of the origin of vertebrates, which was marked by a quadruplication of the entire genome. The expression of Hox genes is highly conserved across animals, and similar genes control similar body segments in different species. In fact, all bilaterians use the same set of Hox genes to control their anterior-posterior patterns, which indicates that they arose from a common ancestor.
When Hox genes become mutated,
The body parts end up developing in the wrong places on the body. When the function of Hox genes increases more than normal, it causes a posteriorization in the affected area, causing complications such as synopolydactyly, which is the fusion of fingers, specifically the ring and the middle finger, such as seen in the slide, or the toes. On the other hand, homeotic transformation occurs when genes lack function, resulting in the replacement of one body part with another, typically in a specific segment of an organism's body plan. For example, as we can see here, the mutation expressed a gene called antennapedia, causing the fruit fly's antennae to develop into legs. Hox genes can turn other genes in our body on or off, kind of like a light switch. I'll be explaining this concept a little bit further in the succeeding slides. The quadruplication of the entire genome is a process in which an organism's entire genome is duplicated, resulting in 4 copies of each gene. This phenomenon is said to have occurred multiple times over the course of evolution, leading to the development of new and more complex organisms. The quadruplication allows for the emergence of new genes and gene functions, as well as the evolution of new species. In some cases, one or more copies of the duplicated genome may become non functional over time, while others may become specialized for new functions. The Amphioxus, also known as lancelets, is a small primitive marine protochordate that is considered a living representative of the ancestral form that gave rise to vertebrates. It has a simple body plan and serves as a useful model organism for studying the evolution of the Hox gene family. Compared to vertebrates, which have four Hox clusters, the Amphioxus only has a single Hox cluster with 15 genes. Their relatively small number of Hox genes compared to vertebrates allows scientists to study the function of each individual Hox gene in detail without the complication of redundant or overlapping functions that can occur in more complex organisms. Now, here are a few terms we should know. So, gene clusters arranged vertically are called A, B, C, and D, as you can see here. As stated before, vertebrates have four gene clusters. That's why we have four letters. Next are the individual genes, which as we can see here are arranged horizontally. It goes from 1 to 13, starting from the 3' end. The individual genes with the same number, for example, A4, B4, C4, and D4, are called paralogs. Be careful not to confuse it with gene clusters. Gene clusters are more general. Basically, their role is to categorize each cluster as A, B, C, and D. Paralogs, however, have to be the same individual gene number, such as seen here, A4, B4, C4, and D4. And then, a set of these paralogs would be called a paralog group. So here's a short animation to visualize how Hox genes work. This is not exactly how it works, but it helps to understand it a little bit. What's basically happening here is the flow of how the Hox genes work by sending signals to other genes, telling them what to do. If a Hox gene is turned on, it can tell a gene to turn on too. And if it's turned off, it can tell a gene to turn off. This is how genes control the development of different body parts and make sure they grow in the right place and in the right way. It follows an anterior-posterior pattern, kind of like dominoes, and it starts from the head at the 3' end and then goes down on a cascade until it reaches the tail end, which is the 5' end. Now, we will be talking a bit about other gene clusters. So Hox genes are present in all the germ layers of the vertebrates. However, for invertebrates, they are limited to the ectoderm. The other germ layers are controlled by other gene clusters. So what are the other gene clusters? The parahox cluster is a sister cluster related to the Hox cluster. It regionalizes the endoderm and contains three genes, the GSX, PDX, and CDX. The GSX gene is particularly important in the development of the brain and spinal cord, as well as in the formation of the pancreas and other organs. The PDX gene is expressed during the early stages of pancreas development and is required for the formation of the pancreatic buds. It is also involved in the differentiation of the pancreatic cells into endocrine and exocrine cells. Lastly, the CDX gene is involved in the regulation of stem cell differentiation in the intestinal lining and expressed in the tail bud region of the developing embryo, controlling the formation of the tail and hindgut. In humans, mutations in the GSX and PDX genes have been linked to a number of developmental disorders and diseases, including diabetes and cancer, while mutations in the CDX gene can lead to abnormal development of the intestine and can range and a range of gastrointestinal abnormalities. The NK gene cluster is a group of homeobox genes that play a role in the regional patterning of the mesoderm. Under the NK gene cluster is the NKX homeobox genes, which forms one gene cluster in Drosophila, and in Amphioxus, this cluster is broken down into three pairs. Then in the vertebrates, it is more dispersed. One of the NKX genes, called the NKX 2.5 gene, or Tin Man in Drosophila, is very involved with the development of the heart. As mentioned earlier, Hox genes must have appeared in the common ancestor of all bilaterians. This includes the NKX 2.5 gene, which is involved in the development of the heart. However, assuming that primordial bilaterans were as small as the organisms found in the Doshanto formation, they probably did not need a heart to keep their tissues oxygenated, since they should have been able to do diffusion. In the Cambrian explosion, the animals became large. Thus, there was a need for a circulatory system since diffusion could not have been enough to oxygenate their tissues. Assuming that the NKX 2.5 gene was present in primordial bilaterians, despite them without having a heart or circulatory system, then the NKX 2.5 gene was probably originally in charge of specifying the anteroventral mesodermal regions instead of only specifically developing the heart. So here's a bit of analogy. Let's say an organism represents a house, and of course, a house needs a team of people to build it. These genes are like different teams of people in charge of different parts of the house. The architect would be the Hox genes, who plan and design the layout of the house. They oversee how the house is coming along and if everything is going according to plan. Next, we have the electricians, which are the GSX genes, and they are involved in the development of the nervous system and have been shown to regulate the differentiation of specific neuronal cell types. Electricians, similarly, work with electrical wiring and connections that facilitate the transmission of electrical signals throughout a house. Next, we have the PDX and CDX as plumbers. As mentioned before, the PDX genes are involved in the development of the digestive system, including the pancreas and liver, while CDX genes play important roles in the development of the intestine and hindgut. These functions are analogous to the work of a plumber in ensuring proper flow and drainage in a house's plumbing system. Lastly, the NKX genes are the interior designers. They are involved in the development of various organs and tissues, including the heart, lungs, and thyroid gland, which contribute to the overall internal architecture of the organism. With all of these teams working together by following a proper rhythm and order, a good and functional house can be made. That is to say, a functional organism can successfully develop. Now to everyone's favorite part of every presentation, the post-test. What do you call individual genes with the same number? For example, A4, B4, C4, and D4. You may pause the video for a bit so that you can have more time to think about it.
And now we go to me. And the question Kuya left for you at the beginning, the answer is dorsal ventral patterning for me. So they have many experiments. So starting with Xenopus, there's B1 organizers who gets BMP inhibitors such as Cordine. It controls the ventral dorsal gradient of BMP signal activity in the animal, aka makes the dorsal ventral patterning of the animal. As you see to the image to the right, the dorsal and ventral portion of the mesoectoderm and endoderm possibly are different for the front and back. This is to specialize the back and the front. For us vertebrates, this will be the spinal regions, that's why it's called neural head notochord muscle, while the front is epidermis, blood, and mesenchyme because that's where the heart is. And now for another experiment, this time on Drosophila. This has another BMP homologue named decapentaplagic, or DPP. This one wants order, while an opponent to this is short tertiolation, which wants disorder. That's why the two men are currently fighting. So for another animal experimented on, which is the planaria. Here we have the analogy of the trolley problem. If it's occurring naturally, the regenerating planarians will have BMP homologs on the back. But if there's RNA knockdown, it will switch the track and express BMP homologs in the ventral portion. And with all those experiments, we now found out that dorsal is in vertebrates, but it's ventral in the invertebrates, so it's opposite. So the reason for this is the principal nerve core is on the dorsal side for the vertebrates, while some invertebrates it is on the ventral side. In the picture below, you can see that it is opposite. The reason for this is like a puzzle piece. You cannot put the same piece in the same side. So you can't put the heart where the spinal cord is and vice versa. And so with all that, what does this tell us? Well, simple. There is a primordial system of dorsal ventral patterning in all animals. But at some point, it became inverted. That's why we have the invertebrate and the vertebrate. Huh? And there's many reasons. So let's start with reason 1. They just felt like it, you know? They didn't have a cord because vertical center inverted the habit. It's just like, I want back spines, you know? And so the other reason is that the rotation of the trunk relative to the head. The trunk spin, but the head stayed. I can't do this, but look, the boneless man and ninja can do it. So this explains why there are nerves, possibly, that does this. That's why it's bent, because originally it was supposed to face the same way. And now to another topic titled sense organs and cell types. So one of the important features of bilaterarian animals is a head, concentration of the sight, sound, taste, smell, and other organs. And now to the PAX6 genes, where it's important to the eye. So we have the mouse where it makes small eyes, as you can see in the picture, and we have Drosophila, which makes eyeless. And now for special case 1 of 2. So here's cephalopods, your very favorite octopus. It's structurally similar to the vertebrae eye. That's one of the things. The difference is the retina is backwards. So for us humans, the retina is at the back, while for the octopus, the retina is in the front. They also don't have a blind spot that way because of that. It's analogous because it's hard to believe that such an animal that did this with many trees somehow had the same idea of the eye, which is very hard to believe. But the answer for this is apparently, because it's the bio-transcription factors. We have homologous, at least to some extent, for the PAX genes, which is eyes. And so we have special case 2 of 2. So we have the planarian, our favorite lovable animal that we never harm. And now this time they cut it once again to see which gene induces eye development. And they found out PAX6 is not eye development for planarians. And it apparently needs cynocolis, which is what they require. So for Drosophila, cynocolis is the most important because it basically downstreams PAX6, aka if you can't win the competition, kill the competition. That's what cynocolis did. It killed PAX6 so that it's the only one that can influence the eye. So in mammals, the development of the eye is just 6'3", which is also another homolog. And after all that, why is this important? Simple. It showed that there is a remarkable degree of conservation for a group transcription factor for the creation of a light-sensitive organ, aka the eye is apparently there for almost every animal in the animal kingdom. It's just that some of them just lost the eye or chose to get rid of it. But most of the animals have an eye transcription factor, even the eyeless lizards. And now we go to the long-range homology, which is described as individual differentiated cells and the gene responsible for their formation, aka it's very few cells that does a lot of things, aka it's a multitool. We have long-range homology for it is very apparent by Bilateria, and for sponge and diploblasts, there's small angular cells with multiple functions, like I multi-told earlier. So their sponges have light-sensitive steering rudder cells, basically light-sensitive like the eye, and steering rudder cells to steer. They have the myoepithelial, the muscle skin of the Darians, so it's like an understaffed kitchen. They have multiple functions. And now we have the MyoD genes, which is myoblast determination protein 1, that is the actual name. It is prototype of the myogenic family. They exist in all bilaterians, and they are important for differentiation of muscle cells. Just remember MyoD. Myo is myosin, which is muscle, and D, differentiation. You need that for your exams. And now we have a few more genes besides MyoD. We have opsin and delta notch signaling system. Opsin is needed for photoreceptors, so opsin sounds like optic, which is eye, and delta notch signaling is like delta, which is area and military, and notch is like a small notch, like a switch that can change spots, which is needed for neurons from neurogenic epithelia, like connecting neurons with the switch. And now we go to basal animals, which is the last subtopic for me. Basal animals are like the base animals, they're very simple. So for here to the right, we saw evolution can give me scales for protection. The protection, yee. And apparently it used it to create wings, to create a way to heat up and other things. So with all the things that we went before, all genes have been sequenced, transcription factors, we also show that we now know what genes are needed to make an animal and when they arose in evolution. Some of the families of development has control genes. So encoding transcription factor and signaling systems, they are found in quanoflagellate, which yes, it looks like sperm. Yes, it's a small sperm. No, it's not a sperm. It's a very small animal because most of the system of animals have Hox genes or some other Hombox gene, ones or TGF beta S, and nuclear hormone receptor. They appear first among the animals, but for quanoflagellates, they have encoding transcription factors and signaling systems. And now we go to the most basal animals and modern phylogenies, the porifera or the sponges. And because they're so basic, they're the one taught first in Inverzoa and Inverzoa lab because of how simple they are. They are thought to have very few cell types and no real tissue grade organization. They only contain NK-like clusters of Hombox genes, but no true Hombox genes. And now another basal animal, the Placozoans, which also has no Hox gene and no tissue grade organization. So they're basically just like sponges, but it looks even less like an animal because it's so small. As you can see, it's barely different from a stain on a glass. And now we have another type of basal animals, the Cnidarians, which composes of the sea anemones, the jellyfish, and the hydra. And in special case, the hydra is used for regeneration research. And special case for Cnidarians, why? Well, they are basal animals, but they have two germ layers rather than three. So they have the ectoderm and the endoderm. And they are radially rather than bilaterally symmetrical, which is also different than the other animals. They do have Hox genes, so they're very closer to real animals, but they do not form linked clusters. Rather, they are scattered singly or in pair, which make them very unique. And now we have the sea anemone, specifically the Nematostella. They have been domesticated for lab work, so it can be reared in the lab, of course. It can do its complete life cycle and have a face of mobility and sessile, as you can see in the right side. And they're specifically done for regenerative work. And now we go back here. So we know now? We sure? We know what we're doing? And no, we're still not sure because why? There's little staggering or expression limits, and it's difficult to establish if anything even does anterior posterior in bilaterians. Like look, here we have the vertebrae, arthropod, and anemone. Do you see the problem? Anemone barely has BG3 and central genes related to the others, and for the posterior it barely has any other genes besides D and E. So it makes it hard to correlate. Even worse, Cnidarians are not even completely radially symmetrical. Some of them could be bilaterally symmetrical or no symmetry at all. So it makes it very hard to do and confirm anything. And you want it to be even more confusing. Look, there's a study of BMP and chordine homologs in Nematostella. The problem here, BMP and chordine are both expressed on the same side of the polyp in E. Look to the right. Yes, it's on the same side being expressed, making it unclear whether it's actually a directive to the axis or it's just whatever it wants it to be. And to add con to the fusion to confusion, we have the Cnidarians may not really be diploblastic, so that makes it even worse. So now the confusion continues with the jellyfish has striped muscle cells specifically in podocorine carnea. They can do a complete life cycle in the laboratory. Okay, that makes it simple. No, the polyps have asexual buds which produce medusae. Medusae does sexual reproduction to create a multiplanula larva, which then cells become new polyps, which is a sexual once again. The cycle repeats. Now, why is this confusing? Okay, tell me, are the jellyfish sexual or asexual? You're about to answer both, aren't you? Yes. That is why it's also confusing what kind of animal they are. The mother cells of the medusae develop from a cell layer called the entocodon, which arises in the asexual buds and expresses the mesoderm type gene, including the homologs of twist, snail, and MEF2. Yes, if you look at the picture at the bottom, there's a colony that releases a bud that creates a mesoderm-like format. As you can see in the stage 2 and stage 3 to 4, there's like a mesoderm-like system covering it. And now there are also Cnidarians without medusae, but they still have the same genes that express and reach the endoderm, which then gives rise to the suggestion that the mesoderm originates as a subdivision of the endoderm, because it's still there even if such a thing should not have existed. And so the study of basal animals has shown that most of the genes needed to make an animal were present when the Neanderthals originated. On their own, these genes do not lead to bilateral body plan, but once such a body plan arose with various development control genes co-opted into setting up its key features, the head, this, everything, and they were like, this thing works, let's continue doing that. And then it led to long-range homologies, the multi-tool, and it can be detected today by genomic sequencing and expression analysis. And now that ends my part, let's go to Mr. Valmores. Hello, I am Mr. Valmores. Mr. Patosa has left a question for you. What gene is present in all vegetarian animals that is needed to differentiate muscle cells? It is 4 letters. He left a hint for you. This is related to actin myosin in terms of naming, and 1 letter is already present in the question. And here is a graph to help you with the answer. Thank you. Have you already have enough time? Mr. Partosa says there is already enough time. It's been 30 seconds. The answer is MYOD. MYOD.
What really happened in evolution? The other main area where developmental biology can assist in the solution of evolutionary problems is to find out what actually happened in evolution. Some evidence about this can be obtained by comparing the expression patterns of key developmental genes that gives rise to 2 different morphologies, one which is the ancestral and one which is derived. Elucidating developmental mechanisms, it is also necessary to establish the actual biological activity of the genes in question by performing overexpression and loss-of-function experiments. This may sometimes be difficult as the organism under study will not be the standard laboratory model, and so it may even be difficult to obtain embryos, let alone perform successful experiments on them. When trying to decide what happened, it is important to avoid the fallacy of design. In other words, once a developmental system is understood, there is a tendency to say extra legs can be produced very easily by allowing gene A to turn on gene B. So this must be what happened in evolution. Firstly, it may not have happened. Maybe the extra legs were formed without A turning on B. Second, even if A does turn on B in the new position, the mutations that cause this to happen may not be in A or either B. They may be in different genes altogether, and there may be multiple small changes rather than one large one. So in general, because they have arisen by natural selection, developmental systems are much more complex that would seem necessary had they been designed by a conscious agent, and they do not necessarily change from one behavior to the another. This consideration also relates to the issue on the developmental constraints. So biologists not concerned with developmental mechanisms often implicitly assume that there are no limits to possible mutational variation, and with enough mutants, enough time, and enough selection, it is possible to evolve anything from anything else. I have here an illustration which is the developmental gene expression in the anemone Nematostella. So as what you can see on the figure A, it is the Nematostella polyp scale bar which is measured by 1 centimeter. B, we have the planula larva scale bar of 70 millimeters. On the figure C, we have the primary polyp scale bar which is 80 millimeters. Uh, in D and E, we have in the planula, chordine, and decapentaplegic are expressed on the same side of the body. So asterisks marks the blastopore, and we have in figure F the comparison of Nematostella hox genes with those of vertebrate and Drosophila. And on the G, or the figure G, we have the expression of hox genes in primary polyp. Developmental biologists often tend to fall into the other extreme of believing that only mutational variation that is possible is that produced by gain or loss of function of each known component of a particular developmental system. So with this, this will confine the range of attainable morphologies to a relatively small set obtained by one-step changes away from the wild-type pattern. So mutagenesis screens on vertebrates such as the zebrafish or even the mouse generate only a modest range of its phenotypes. The reason is that mutations in pleiotropic genes with multiple functions are often early lethals, and also that many morphological changes are not seen because they would need several mutations to present simultaneously. The potential importance of developmental constraints to evolutionary theory is immense. For example, if constraints are very limited, then the course of evolution is determined almost entirely by adaptation to a changing environment. But if the developmental constraints are substantial, then the course of evolution is determined mostly by what is possible given the current situation. So at present time either, the true extent and role of developmental constraints remains unknown. So in general, the mutations of the unknown components of well-understood systems, such as those controlling segmental identity or limb patterning, will be macromutations. So single mutations which bring about a significant morphological change in one step. So neo-Darwinists have always been uncomfortable with macromutations, believing that they will virtually always be deleterious and select against. So we have this developmental biologists are often more sympathetic to what has been called the hopeful monster, as what you can see on this slide. So it's the creature arising from a macromutation because this type of mutational change is congruent with developmental explanations. Also, some morphological changes characters seem intrinsically qualitative, and it is hard to imagine small, imperceptible intermediate forms.
Next is we're going to discuss the segmented body plans and Hox genes. So first, let's have a recall. Segmentation is a fundamental feature of many animal body plans, where the body is divided into repeating segments that may be specialized for different functions. So the development of segmented body plans is controlled by a group of genes called Hox genes. So at what was previously discussed, Hox genes are responsible for specifying the identity of each segment in the developing embryo, since they encode the transcription factors as well as the proteins that regulates the expression of other genes. So the segmented body plans are found in many different groups of animals. So for instance, we can see these three different animals here, such as flies with 6 legs, mice with four, and the centipedes which has many legs. So as you can see, they have a varying body plans. In Drosophila, QBX or ultrabithorax and ABBA or abdominal A determines the boundary between thorax and abdomen. QBX specifies the identity and development of the third thoracic segment and the first abdominal segment. On the other hand, ABBA expressed in the posterior abdominal segment of the thigh. So, as what we can see, there is an association of Hox domains with body regions, but there is no specific association with the thorax or with the suppression of legs. As the previous example, which is the Drosophila, the Hox gene is expressed in the abdomen, as shown in the first figure, which shows the ends of segmented body plan. In the crustaceans, these two Hox genes expressed throughout the thoracic segments, all of which bear legs but are not expressed in the head or genital regions. While in the third figure, which is the myriapods, QBX and ABDA are expressed in all but the first thoracic segment. And lastly, in the chelicerates, the anterior boundary occurs within the ophistosoma, the posterior body region, which does not bear any appendages. So these are some association of Hox boundaries with boundaries of segment type, but the specific functions such as leg suppression are still not conserved. In the first figure A, we can observe the wild-type hatchling showing maxillipeds in fin. In the second figure B, Reduced UBX protein synthesis results to T2 and 3 appendages develop as maxillipeds. And overexpressed UBX in the third figure causes a variety of pasteurization in the head and thorax, including conversion of antennae, maxillae, and maxillipids into thoracic lights. So we can see that UBX does not control appendage character in this organism, but the nature of the mutation that changed the expression of the UBX.
Now, we will proceed with the vertebrate limbs. So, the vertebrate limb has a distinguished history in evolutionary biology. So, it was early recognized as being a homologous structure across the tetrapods. Because of the presence of a complex skeleton, it is often preserved in fossils and it provides some striking examples of allometric changes in evolution. Means differential growth between parts. So for example, the middle digit of the horse leg has elongated during evolution until the modern horse has effectively just one digit with the others reduced to vestiges. So by contrast, all digits of the bat forelimb have become greatly elongated to bear the skin flaps that constitute the wings. So I have here an illustration of the origin of the leg. So take note that this illustration will just serve as an overview on what are you going to expect on the succeeding slides. So on the letter A, we have the Panderichthys and its four-limb skeletons. B, we have the zebrafish pectoral fin skeleton, and on the letter C, we have the Ichthyostega with its hind limb skeleton. Do note that on the succeeding slides, each one of these will be discussed thoroughly. We have here the Panderichthys and its four-limbed skeletons. So it is believed from the fossil record that the first tetrapods arose in the Devonian period and that their ancestors resembled a creature called Panderichthys. So this was a lobe-finned fish belonging to the same order as the modern coelacanth. It is thought to have lived in shallow water and perhaps even be able to creep out of the water like some of the modern catfish. So this fact was associated with the existence in tetrapods, but not in teleost fish of an enhancer for the HOXG13 gene, which caused late expression in the region destined to become the autopodium. The idea arose that the origin of this genetic element was the key to the evolution of the tetrapod limb. However, it is now known from computed tomography scanning of Panderichthys fossils that the organism did, in fact, have distal skeletal elements resembling a primitive autopodium. So with this, I have an illustration about the zebrafish pectoral skeleton. So the study of development of the paired fins of zebrafish have provided further evidence for common developmental mechanisms underlying their different anatomy. So the injection of morpholinus against the WNT2B or the TBX5 will both inhibit fin bud formation as in higher vertebrates. So during the early fin bud outgrowth, there is an apical ectodermal ridge expressing FGF genes and a posterior region expressing SHH, which is very similar to the tetrapod limb bud, but during this phase are laid down four parallel cartilage elements, which later ossify and serve as the attachments for the fin rays. Both the pattern of these proximal bones and the fin rays themselves, which are formed by dermal ossification, are quite unlike a modern tetrapod limb or even the Panderichthys limb. One of the most obvious features of the modern tetrapod limbs is that the number of digits very rarely exceeds 5. Again, very rarely exceeds 5. The very name pentadactyl limb indicates that the primitive limb has 5 digits from the word penta, and that all types with reduced digits are derived. It has been speculated that there is a developmental constraint preventing more than 5 digits because this is the number of Hox-G genes available to create distinctions between the digit territories. Unfortunately, this theory does not stand up to the fossil evidence because there are some early Devonian tetrapods, such as the Ichthyostega, appearing soon after Panderichthys, having an autopodium which is very obviously possessed more than 5 digits. How Ichthyostega achieved this is still not known. Let's proceed to the limb presence and positioning. As what you can see on the slide on the left portion, I have here an illustration about the Hox gene expression in the flank of cheek and python embryos. So some tetrapods have greatly reduced limbs or have lost them altogether. This includes the snakes, the whales, and the forelimbs of the flightless birds. Often a rudimentary girdle or portion of the proximal limb skeleton survives as a vestige, indicating that the ancestors had more substantial limbs than their modern descendants. The positions of the limbs on the lateral plate must be specified by the antero-posterior patterning system for the whole body. So evidence of a role for Hox genes or Hox gene is provided by the effects of transgenic expression of the HOXB8 at a more anterior level than the usual in the mouse, which results in the formation of an extrazone of polarizing activity or the ZPA and duplication of the forelimb. In the mouse and chick, the Hox genes C6 and C8 are expressed in the lateral plate between the two limb buds, and it is believed that they repress limb formation in this region. The python is a type of snake that has no trace remaining of the forelimbs but does have a rudimentary pelvic girdle and a proximal vestige of the hind limbs. It shows expression of the hocks C6 and C8 extending right up to the head, but posteriorly the expression border stops just short of the rudimentary hind limbs. Analogy time. So to sum up, or to sum up with the key important um, notes about my report, as what you can see on the slide, we have the concept of natural selection, particularly in evolution. So evolution affects or it has a greater or a great impact in the development of the organisms, particularly in their physical features. So by natural selection, developmental systems are much more complex than would seem necessary had they been designed by a conscious agent, and they do not necessarily change from one behavior to another using the most simple route. So, in short, evolution has a great impact to the developmental process of certain organisms. With this, evolution, which is another branch of science, and developmental biology are interconnected with each other.